can you see the screen?
Yes, yes,
Yes, sir. Yes.
Okay, all right. So good evening, everyone. I'd like to talk on the molecular diagnostics in cancer with a focus on hematopathology. Okay, so before we start about molecular diagnostics, I'm going to do a bit of introduction to the human genome. So if you see the human genome, these are chromosomes which are present, which all of us have 22 chromosomes and in pairs and an X and Y chromosome. If you look at this structure of the chromosome in great detail, you can see that each of it is composed of this DNA strand, and this DNA strand is actually in a double helix format. So you have an alpha strand and a beta strand. Sometimes this is called as a Watson strand and a Crick strand. But for representation of the genome, this is represented in the reference sequence as one straight line, which is typically from 5' to 3'. And it is because the other strand can always be inferred if you know the sequence of this particular strand. So there are some terminologies which are used in the parlance of molecular diagnostics which you should be familiar with. Well, first is locus. The locus is segment of DNA which occupies a particular position or location on a chromosome. So locus can be large, for example, many genes, for example, an HLA locus, or it can be small, for example, beta globin gene locus. So you can see here that this is the locus for coat length hair, and this is the locus for coat color. So this is like the coat or the hair of the dog. This is how long his hair should be, how short his hair should be. And allele contains alternative versions of the DNA sequence in a locus. So you can have an allele for short hair, an allele for long hair, or similarly for white fur or black fur. And there are also some terminologies that one comes across like wild type and mutant. So wild type is the single prevailing allele which is present in more than half the number of individuals in a population. It is known as a wild type or common allele. Other alternative thing is when the wild type allele is altered or changed, it is known as a variant or mutant allele because it differs from the wild type allele because of the presence of a genomic variation or mutation. If a particular change is there, that is, if there are two or more common alleles at a frequency of more than 1%, this particular locus exhibits a polymorphism or many forms in a population. But as you know that Indians as well as many communities marry amongst their own communities or amongst their own caste or religion or whatever, and they can have alleles which are then replicated in their particular population, and that is called as variations which are not frequent enough in a particular population, and they are known as private alleles. Similarly, there is a concept called as haplotype, which is a set of DNA variations such as polymorphisms which are adjacent to one another that are inherited together, and these alleles are known as linked polymorphisms. So if you have a particular set of alleles here as A, G, and T, as well as another set of alleles for A, G, T, and A here, as well as a third set which is A, G, and A, these are all different haplotypes for the same region. So mutation is a permanent change in nucleotide sequence or arrangement of DNA. Chromosome mutations are they can occur at a gross chromosomal level, where the number of chromosomes has changed. You can have regional or subchromosomal mutations, for example, a large deletion or a duplication. You can have gene level or DNA mutations, which are substitutions, insertions, or deletions of DNA, arbitrarily set to a limit of 100 kb. So if you look at genomic alterations, they can be classified as microscopic, which are identified through chromosome binding techniques, and account for at least 500 kb of DNA because they have to be seen through a microscope. And then you have submicroscopic variations, which are identified through more sensitive techniques like RACGH or NGS. So this is an example of a whole chromosome aneuploidy, for example, trisomy 21, where there is a gain of a particular chromosome. Similarly, you have a loss of a particular chromosome that can take place as well. These are examples which are seen through chromosomes. Also, similarly, you have segmental aneuploidies where you have deletion of part of chromosome, translocation of part of another chromosome, or duplication of part of a chromosome. Structural rearrangements are classified also as balanced if there is an transfer of genetic material from one chromosome to another, and unbalanced if there is additional or missing material. Unidentified chromosomes can also be seen, and these are known as marker chromosomes, and typically are a hallmark of complex chromosomal rearrangement seen in cancer, especially highly growing or rapidly growing cancer. So deletions can be terminal deletions at the end of a chromosome, or they can be interstitial, which occur in between. So for you can also have other structural abnormalities like ring chromosomes or isochromosomes. A ring chromosome occurs when both ends are gone and these ends fuse to each other. Isochromosome is seen when one entire arm of a chromosome goes missing and then gets duplicated in a mirror image fashion. You can similarly have reciprocal gene rearrangements in which there is exchange of genetic material between two chromosomes, for example, 9 and 22. And sometimes you can have Robertsonian translocations, where translocations take place at involving two acrocentric chromosomes which fuse near the centromere region. There are also some other types of gene mutations, and these are typically detected through techniques such as Sanger sequencing or next generation sequencing. So here, let us say you have one hypothetical gene where there is a TCA which encodes for one codon. Here you can see that the T changes to a G mutation, and the TCA which codes for serine now gets changed to GCA which codes for alanine. You can also similarly have deletions or insertions in the amino acid reading frame. Here, instead of a TCA, the T gets deleted, and therefore the reading frame shifts by one nucleotide towards the left, as a result of which the entire coding sequence downstream of this deletion or insertion gets changed. So for example, to give this example, I love Guns N' Roses in a rock concert. Sure enough, it's a legitimate statement. But if you delete a part of the sentence, not only does the sentence change, for example, I love guns at a rock concert, but the entire meaning of the sentence also changes. So this is how insertions and deletions cause change in the amino acid reading frame. This frame shift results in different sequence of codons, and which result in therefore a functionally altered protein product. There are other type of mutations which occur at a submicroscopic level. These are again detected through techniques such as next generation sequencing. For example, deletion of a particular region of the chromosome, a duplication type of copy number alteration, and an inversion wherein an entire segment of the genome gets inverted. So what are the applications of molecular diagnostics to hematopathology? First is to diagnose diseases which are defined by a molecular abnormality. Then to support the diagnosis of a disease, to support the diagnosis of a malignancy by demonstrating clonality, to predict prognosis of a disease, to decide the treatment of a disease, to monitor the response of therapy, and to determine cause of suboptimal response to therapy. And lastly, in identifying inherited blood cancers. So let us see some of the examples which are there. First is diagnose diseases defined by a molecular abnormality. So as any undergraduate or at least an MD pathology student can tell you that this picture is not normal. There is obvious leukocytosis here with an increase in myeloid cells in the peripheral blood film, as well as increase in eosinophils and basophils. And this patient most likely has a myeloproliferative neoplasm, which is likely to be CML. CML is actually the poster boy of molecular medicine. And you can see that in the early 60s or early 80s, you had a single digit survival, long-term survival for CML, which was increased, which was improved by bone marrow transplantation as well as other techniques. But it was actually the introduction of chemotherapy-free drug, which is imatinib mesylate, which would transform the treatment of AML, because you now had a single abnormality which could be targeted by a specific inhibitor. So CML, as you know, has a molecular definition which requires the demonstration of the translocation 922 or its product, the BCR-ABL1 fusion gene. The BCR-ABL chimeric protein is integral to CML leukemogenesis, and blocking the ABL kinase using tyrosine kinase inhibitor results in regression of CML and durable clinical responses in nearly all patients. And the standard of treatment now is daily lifetime therapy with imatinib mesylate or its newer drugs. So as we know, the genetic basis of CML is a translocation between chromosomes 9 and 22, which results in a derivative chromosome with the BCR-ABL fusion protein. This is typically diagnosed by techniques such as conventional karyotyping and FISH, and as well as can be diagnosed by techniques such as real-time PCR. So the advantages of real-time PCR or PCR-based techniques is that this is a highly sensitive technique as compared to FISH and conventional karyotyping, and can distinguish between different types of BCR-ABL transcripts. So whenever a new patient of CML comes to our laboratory in our hospital, we do testing by FISH as well as send a sample for transcript identification, which helps us identify what is the type of transcript the patient has. The disadvantage is that this is an RNA-based test, and this is a test that is susceptible to RNA degradation, and this can lead to varying reports between the labs unless the test is standardized very properly. And all these tests, for example, P90 kilodalton CML transcript is there, P210 transcript is there, all of them require different PCRs and different real-time PCRs. And the main use of PCR in CML is actually monitoring the copy numbers of disease after giving imatinib. Which brings us to the question, why does one do RNA-based tests? So if you look at the distance between two exons that is there in a hypothetical gene, typically these exons are separated by thousands of bases between introns. And if you were to amplify this particular region of the genome, you would end up need to amplify a very large region which is here. If you were to work on RNA alone, it becomes a very small PCR test, which is hardly of 500 bases, and PCRs which are smaller in size are very easy to perform, as can be seen here. So which is why one works on RNA when you want to detect fusion transcripts, because fusion transcripts, typically the chromosomal translocations or at DNA level happen between introns, and it is very difficult to amplify this such a large region of the genome, especially when you want to monitor responses to therapy. Therefore, if you work on RNA-based tests, these are much smaller regions to amplify and are easier to work on. But RNA is single-stranded, and it must be converted to double, and it is unstable and must be converted to double-stranded complementary DNA, which is done by an enzyme reverse transcriptase. So this is an example of transcript identification in a newly diagnosed patient of CML. You can see here that if you do these real-time PCRs, you get amplicons of a different length. So depending on how your assay is standardized, you can have E14A2, E13A2, and E1A2. All these are translocations which are of different types in CML. The E13 and E14A2 are typically seen in CML, whereas the E1A2 is typically seen in Ph positive ALL. The correct acronym for this is qPCR, and RT-PCR is actually reverse transcription PCR. So this is an example that most of you would be familiar with. Here you can see that the patient has abnormal promyelocytes or these cells that are there with apple core or this figure of 8 nuclei and intensely granular azerophilic cytoplasm, which when stained with MPO results in an intense reaction. And this is typical diagnosis of acute promyelocytic leukemia. So acute promyelocytic leukemia is defined by translocation 1517, which is detected by FISH. As you can see, this translocation between 15 and 17. If it is yellow, it indicates that there is a fusion transcript, which is here. But if you see this at RNA level, RT-PCR is required to identify the type of fusion transcript, because there are three common fusion transcripts which are seen in PML-RARA, which is the BCR1, BCR2, and the BCR3 isoforms. And once you identify which isoform is there in the patient, you can do breakpoint-specific qPCR to follow this patient up. So as I said, the first application of molecular testing to hematopathology is to diagnose a disease which is defined by that specific molecular abnormality like CML and acute promyelocytic leukemia. The second is to support the diagnosis of disease. Now let us see this example. You have a 69-year-old female with dyspnea on exertion, easy fatigability, and the patient has got anemia and neutropenia. Cytogenetics is normal and flow cytometry is negative, and this patient has got mild dyserythropoiesis here, but there is no definitive diagnosis of MDS on morphology as well as flow cytometry. But when we perform next generation sequencing for this patient, you could see that this patient has a missense mutation in exon 14 of SF3B1 gene at a variant allele frequency of 17.4%. And if we went back to this patient, performed an ion stain, and you can see the presence of these ring sideroblasts which are there. So this is an example of an SF3B1 associated myelodysplastic syndrome. And you can see that SF3B1 mutations are very commonly seen in myelodysplastic syndromes. They are the commonest type of mutations in myelodysplastic syndromes. They typically occur at the A666 hotspot, but can be seen in other locations of the SF3B1 gene as well. This will result in changes in diagnostic criteria for MDS. This is the older 2017 version of the NCCN, where instead of ring sideroblast, even if the SF3B1 mutation is there, it was okay. Today, the diagnosis in the WHO can just be that you have a patient with low blast and SF3B1 mutation, and it can be diagnosed, and that itself is enough to diagnose MDS, irrespective of whether the patient has ring sideroblast or not. Similarly, you have diseases such as this, which is the example of hairy cell leukemia, or this where you have plasma cells and lymphocytes as well as cells which are lymphoplasmacytic in nature, and this is an example of lymphoplasmacytic leukemia. So to diagnose entities where you can have a PCR to confirm the diagnosis of disease, this requires a specific type of PCR known as allele-specific PCR, which answers the question: is this mutation present or not? So when should you use a technique such as allele-specific PCR? You should use this technique when the mutation that you are looking for defines the disease. For example, JAK2V617 in a patient with myeloproliferative neoplasm, or BRAF V600 allele-specific PCR for diagnosing hairy cell leukemia, or MYD882263 to diagnose the presence of lymphoplasmacytic lymphoma. But it must be remembered before you apply such tests that the mutation must be very common in the context of disease. So you can see here, this is a specific PCR, which is used to detect a recurrent mutation such as a SNP or an indirect. This is an example of the BRAF gene, wherein at position 1799, you can see that the GTG is changed to GAG. So the wild type allele is which is changed to a rotated allele, which is A. And if you want to detect this mutation, you can detect this using allele specific PCR. Here you have an outer primer and a wild type specific primer as a reverse specific mutation primer. So when you do a PCR here, you can see on the both the outer amplicons. So you can see here that both the outer amplicons amplify a largest amplicon, which you can see here, as well as if the mutation is present, the primer, outer primer amplifies with the reverse mutant specific primer to result in a medium mutant specific amplicon, and then you have a wild type specific primer and an outer primer, which result in a normal amplicon. So this is what you would get in a normal person, these two amplicons, but in a disease state, you would also get a mutant amplicon. So this is detected by a technique known as allele specific, known as fragment length analysis, where you have a wild type band, a control band, as well as a mutant specific band. Now, you must understand that this BRAF V600 is not just specific for hairy cell leukemia. It is also detected in conditions like melanoma, colorectal carcinoma, myeloma, papillary carcinoma, pilocytic astrocytoma, and rarely even in AML. So the clinical context is important, and it is important because this is used to diagnose disease in context with pathology, and it is a theranostic marker because now you have BRAF V600 inhibitors. The third application of this disease is of molecular testing in hematopathology is to support the diagnosis of malignancy by demonstrating clonality. So this is a 42-year-old lady with anemia and neutropenia, and you can see here that you have a large collection of these lymphocytes with abundant cytoplasm and presence of these prominent azurophilic granules. So to diagnose such conditions, you require T cell clonality, which is essential investigation in diagnosis of TLGL. So in the context of that the patient has anemia, neutropenia, as well as recurrent infections and lymphocytosis, you can see on the peripheral blood film some large granular lymphocytes are there. You then need to perform flow cytometry to show that there is a T cell population clone, and then do a T cell clonality testing to show the presence of an abnormal clone. So what is T cell clonality testing? T cells develop develop from the bone marrow as progenitor T cells, which then migrate to the thymus, and in the thymus they undergo somatic rearrangement of germline sequences. The four T cell TCR genes are TCR delta, gamma, beta, and alpha, and in the germline configuration, delta and beta genes contain variable diversity and joining regions, and the TCR gamma and alpha do not contain the T lymphocytes. As the lymphocytes begin to develop, they undergo these VDJ rearrangements, and this happens in every lymphocytes. So you can see here that in every T lymphocyte these rearrangements happen, wherein the V and the J genes initially join, D and the J genes initially join, followed by the V and the J genes joining. This then results in transcription of this particular rearrangement and to result in the formation of a TCR allele, and this happens for every T lymphocytes. So when you perform TCR gene rearrangements in a normal person like you and me, you get a Gaussian or a bell-shaped curve like this, and in case of a T cell malignancy such as TLGL, you get a monoclonal gene rearrangement, as you can see here. But it is important to note that this has to be taken in the context of appropriate clinical and hematopathology. So clonality, now the definition of clonality has moved on, and we now not only have restricted the definition to TCR and IGH genes, but others as well. So you have a 34-year-old female here with persistent anemia, leukopenia. There is no convincing dysplasia really on morphology. Flow cytometry is normal, and FISH is negative for any abnormality. However, when we performed NGS, we identified a low wave VAF TEC2 mutation which is present in this patient. Which brings us to the next concept of what is CHIP, ICUS, and CICUS. Let us say a patient has persistent cytopenia. You do a bone marrow examination and cytogenetics, and all are normal. This patient then gets diagnosed as idiopathic cytopenia of undetermined significance. At that point, when you perform an NGS for a myeloid mutation panel and you don't get a mutation, the patient then gets diagnosed as non-clonal ICUS. If a mutation is identified, this is a condition called clonal cytopenia of undetermined significance. So there are four different entities here. First is non-clonal ICUS, which lacks any mutations and has very low clinical risk. Then you have an entity called CICUS, wherein the patient has cytopenia and he has the presence of a gene mutation. As this chip continues to stay in the body, it then transforms into low risk MDS and high risk MDS, each of which has a higher risk of transformation to AML. It is important to know of this, and NGS is a very important diagnostic entity in cytopenia, because patients with clonal cytopenia have a significantly higher risk for development of a myeloid neoplasm than those without any evidence of clonality. So this is data from a large number of patients with cytopenia, which shows that if the patients are cytopenic and have more than one mutation, they have a high risk of transformation to CICUS as compared to patients who are cytopenic and don't have a mutation. So CICUS, or clonal cytopenia of undetermined significance, is defined as the presence of a mutation in patients with myeloid neoplasms, one or more mutations in patients which are otherwise detected in myeloid neoplasms at a variant allele fraction of more than 2%. The patient has to have had persistent cytopenia for more than 4 months in one or more of the peripheral blood lineages. The diagnostic criteria of myeloid neoplasms must not be fulfilled, and all other causes of cytopenia and molecular aberrations must be excluded. The risk of transformation to MDS is currently thought to be lower at 50% in presence of single DNMT3 or ASXL1. When dysplastic features or other MDS-related criteria are met, the diagnosis of CICUS then changes to MDS. Mild dysplasia or rare molecular or flow cytometric abnormalities may be seen in this patient. Now, the next use of molecular diagnostics in hemato-oncology is to predict prognosis of disease, and this cannot be shown better than by discussing acute myeloid leukemia. So AML is a disease which results from clonal expansion of myeloid blasts. In AML, other than 1517, the disease outcomes are very heterogeneous, and earlier it was thought to be because of different cytogenetic abnormalities, wherein patients with favorable cytogenetics had good outcome, patients with unfavorable cytogenetics had poorer outcome. So why does a patient with, however, normal cytogenetics have poor outcome, have intermediate outcome? The reason is because of this question could actually be answered over the last few years because of developments in newer technologies such as next generation sequencing. Next generation sequencing was first demonstrated by the application, large-scale application and utility of next-generation sequencing was demonstrated by the Cancer Genome Atlas, wherein they sequenced whole exomes of several different types of common cancers. And we have then the descriptions such as passenger mutations and driver mutations. Passenger mutations were random, non-recurrent mutations which were not involved in causing cancer to develop or progress, and they probably occurred as the cancer developed. Driver mutations or mutations were repeatedly found at high frequency in many samples of the same cancer, even in different types of cancers. For example, what we saw of BRAF V600. These are involved in the causation or progression of cancer, and therefore were thought to refer to as driver genes. Commonly, these had higher VAFs indicating earlier origin. Now, these particular mutations can have different cellular functions depending on the gene that is involved. But as compared to solid cancers, the AML on the whole has a lesser number of mutations, approximately 5 to 12 mutations in the entire genome. However, these mutations again were heterogeneous, and depending on the type of cytogenetic abnormality, these mutations were there. So in intermediate risk AML, there were maximum number of mutations, whereas in patients with core binding factor leukemia, number of mutations were much lower. So subsequently, there were discovery studies which said that there were recurrent gene mutational pathways which were mutated in AML, which then gave rise to the concept that we can only sequence certain limited sets of genes, which led to the development of targeted gene panels. Today, targeted gene panels are used for risk stratification of and diagnosis and prognostication and monitoring of almost every malignancy, and blood cancers is at the forefront of this. So you can see here that in AML, there are a large number of mutations that are there, but typically 3 to 4 mutations are seen in every case. So this is data from our hospital, which shows that FLT3, NPM1, RAS, DNMT3, and WT1 mutations were the most common in our data set. These mutations also occurred in a non-stochastic manner, with some mutations never occurring together, for example, NPM1 mutation never occurring with KIT, and some mutations commonly occurring together always, for example, NPM1 and DNMT3A, IDH2 and FLT3 and DNMT3A, what you can see here. These mutations also occurred at different variant allele fractions, typically with signaling pathway mutations such as KIT occurring at low VAFs, and some other mutations such as DNMT3A, TEC2 occurring typically at much higher variant allele fractions. These mutations also influence the outcome of disease. For example, FLT3 ITDs, WT1 mutations were associated with much inferior outcomes. Also, RUNX1. So this is an example to show how molecular disease or molecular testing is useful to predict prognosis or outcomes in AML. Molecular testing is also useful in deciding the treatment of disease. So this is an example of a 58-year-old male with leukocytosis and eosinophilia. When you perform the bone marrow examination of these patients, you can see that there are abnormal megakaryocytes as well as the presence of these eosinophilic precursors. So you can see abnormal megakaryocytes here, these basophils, as well as different eosinophilic precursors. So how does one work up cases with unexplained eosinophilia? The first thing to do is to rule out or work up all the reactive causes that are here, such as parasitic infections, allergies, pulmonary diseases, skin diseases, and other malignancies. If those are ruled out, then if the patient has persistent eosinophilia for more than 6 months, if hematological malignancy and abnormal T cell populations are excluded, if the end organ damage is there, patient is diagnosed to have idiopathic hypereosinophilic syndrome. If there is no end organ damage, the diagnosis is idiopathic hypereosinophilia. But you must first demonstrate clonality to exclude certain disorders. So, for example, in a patient with eosinophilia, if a patient has monocytosis, the diagnosis is CMML. If the patient has granulocytic dysplasia, the diagnosis is atypical CML. And then you must rule out other types of fusion abnormalities that are there. So in this patient with eosinophilia, there was an extra copy of ABL1, what you can see here. And this patient was negative for all the BCR-ABL fusion transcripts, as you can see in this particular case. So when RNA sequencing was performed, we identified an ETV6-ABL fusion as a result of translocation 912, as you can see here. ETV6-ABL1 fusion is known to respond to TKI therapy. So this patient, it is rare, with less than 30 cases being reported when we had diagnosed this entity. This patient was started with TKI and imatinib bisalate, and after 3 months, this patient was in complete morphological remission. So examples of therapy decisions based on molecular testing in blood cancers are tyrosine kinase inhibitors for BCR-ABL like ALL, CML, Ph positive ALL, JAK inhibitors for BCR-ABL1 like ALL, polycythemia vera, BRAF inhibitors for hairy cell leukemia, IDH inhibitors for IDH mutated AML, and FLT3 inhibitors for FLT3 mutated AML. Lastly, you can also use molecular testing to monitor response to therapy, and this is shown by the classic example of CML, wherein a lot of molecular testing is done for seeing how the response to chemotherapy is taking, for response to imatinib therapy is taking place. So here, when you want to monitor disease, you take peripheral blood, extract RNA, convert to cDNA, and you have to do two PCRs. One is a qPCR for BCR-ABL, which is a target gene, and one is a qPCR for ABL, which is a control gene. And the ratio of these two numbers is multiplied by 100, use your normalized copy number percentage. So why is this done? Is because 30% of patients with CML have to stop imatinib due to intolerance and resistance. If the patients are treated with CML and they have adequate response, which is sustained, and the patient becomes negative, you can think of stopping the drug. If the patient has suboptimal responses, as you can see by your real-time PCR, you can increase the dose, you can do second-line treatment with TKI, or you can refer the patient to BMT, depending on the type of mutation that you see in the type of resistance. So there are different response definitions in CML, as has been defined by the European Leukemia Network. And you can see here that that typically at a level of 1%, the patient is said to have a complete cytogenetic response. At 0.1, major molecular response, and at 0.01%, the patient has deep molecular response. So if you look at different techniques, you can see that conventional cytogenetics and FISH have a very low sensitivity, and to really detect disease at a much lower level, you need to do sensitive techniques such as quantitative real-time PCR. So what is real-time PCR? Real-time PCR is just like PCR, where you have a forward primer and a reverse primer, but you also have a fluorescent, you have a probe that sits in between the forward and reverse primer. This forward and reverse primer, if the probe binds to the specific target, the Taq polymerase separates the reporter dye from the quencher dye, which is present on the probe, and once the quencher dye gets separated from this gets hit by laser induced of fluorescence. The intensity of the cycle at which fluorescence appears is directly proportional to the amount of transcript or target being detected. When this helps you to derive the CT value from which a number is calculated. So how do we get a number from a real-time PCR CT value? This is done by a technique known as either by absolute quantification or by relative quantification. In absolute quantification, the PCR signal to copy number is detected by using a calibration curve, which are either linearized plasmids or spike tissue samples or whatever that is there. So in our lab, we created this by doing some simple genetic cloning, wherein you take the clone, insert it in the plasmid, insert it into the bacterium, and let it grow on a colony. And once you get these plates, you take these plates and extract DNA from them, and these contain your BCR-ABL transcript. Now you can create a serial dilution of these plasmids and then run your test sample. And since we know what is there, what is the amount of target DNA in this serial dilution, we can estimate an unknown samples BCR-ABL value as well. But as I said before, you also require control gene to normalize this, because as an inherent property, qPCR suffers from problems caused by error in numerous processes. So normalization helps in control for variation in the total mass of RNA analyzed, corrects for any biological or technical variability, and prevents over or underestimating two expression levels. So let me give you an example. So in a patient with BCR-ABL who is newly diagnosed, you have 100 copies of BCR-ABL and 100 copies of ABL. And in another patient, you have which is a follow-up patient or sample, wherein the number of BCR-ABL copies are 100, but ABL copies are 40,000. So you can see here that these are not the same amount of same meaning. Although the BCR-ABL copies here are there, since the ABL copies are very low, you can say that this RNA is completely degraded, and that this is a different meaning as opposed to a follow-up patient with 40,000 ABL copies and 100 BCR-ABL copies. So reference genes are used for normalization, which are done for using genes such as GUS, GADPH, and ABL1. So this is how normalized copy number is done. It is done by mean copy number of the target gene by the mean copy number of ABL1 gene into 100, which gives you normalized copy number. You can also use certain advanced techniques such as next generation sequencing for monitoring this response treatment in malignancies such as AML. So I'm not going to go into this. This is not really required here. But what is important to know is that sometimes you can use molecular testing to find out what is the cause of suboptimal response to targeted therapy. So now for the same hypothetical CML patient, the patient, once you start treating him with BCR-ABL, you can see that the normalized copy number falls and then suddenly starts to rise rapidly over the period of 1 year. So why does this occur? Is because the body or the cancer fights back by developing mutations at the site of imatinib mesylate. So imatinib mesylate as a drug goes and lodges itself at the ATP binding protein of the BCR-ABL molecule of protein, and as a result of which downstream phosphorylation of substrates cannot take place. So here in an untreated state, you can see that the BCR-ABL protein provides this ATP molecule and results in phosphorylation of downstream substrates. But the imatinib mesylate goes and blocks this phosphorylation, and you can therefore this uncontrolled phosphorylation of substrates will stop, reversing the oncogenic ability. So the cancer fights back by developing mutations at the site where the imatinib mesylate is supposed to come and act, and these mutations are so many have been described, and these mutations are then decide what is the type of therapy that can be given. So if you have some mutations like T315I, no drug is going to act, especially ones such as imatinib, dasatinib, nilotinib. You can give special drugs such as bosutinib or take the patient up for bone marrow transplantation. So in this, you take the patient's BCR-ABL, you amplify the BCR-ABL molecule, and then amplify that for that TCR reaction. You do another PCR reaction to amplify the kinase domain, and you sequence it. And you can identify these mutations. Earlier we used to do standard sequencing, but now we do next generation sequencing for this. And this particular patient example here has the T315I mutation. This patient is resistant to all kinase inhibitors and now needs bone marrow transplantation. So last example that I'm going to talk about is a 2-year-old with leukemia who says that his grandfather also had cancer. Are these events related? So one of the last uses of sorry for that. So one of the uses of molecular testing is identifying inherited myeloid cancers. So this is a newly recognized entity by the WHO known as hereditary myeloid malignancy syndromes, wherein patients have mutations in certain genes such as TP53 or GATA2 or RUNX1, ETV6 with a variety of different presentations with different cancers or with recurrent infections or with cutaneous warts or with other syndromes that can be seen here, and these are known as HSMS or hereditary myeloid malignancy syndrome. So you can see a 45-year-old mechanic with pancytopenia has got AML here, and you can see that there is fish shows a deletion 17p in 70% of cells, deletion 5q in 95% of cells, but this patient has multiple DDX41 mutations. One is a DDX41 mutation at high VAF and another mutation at low VAF, as well as a TP53 mutation. So this is now recognized as a new independent WHO entity, which is myeloid neoplasm with germline DDX41 mutations. We have now seen approximately 15 of such patients, 15 or 20 of such patients. They have male predominance, predominantly present in the 6th decade of life, and they have they are presenting AML as well as MDS, and they don't have any other history or features to raise suspicion of this diagnosis. Almost half of the patients have multiple DDX41 mutations. So these are patients who have a somatic mutation as well as a germline or mutation in DDX41, and they can have other mutations such as TP53, ASXL1, and JAK2 as seen in our case. So they are present either at the N terminal or at the C terminal. I'm not going to talk about this. They can also have other concomitant abnormalities such as deletion 5q as is seen in our case. Why it is important is because this has direct consequence on clinical management such as transplant for DDX41 mutation carriers. So for example, if a child or if a son is to donate for his father's AML, he should be screened for DDX41. They should be testing for appropriate family members as well as routine surveillance. So with that, I'd like to say thank you and say that we have a 2-year hematopathology fellowship, and if you are interested, you can check out our website tmc.gov.in if you want to learn more about these malignancies and how you can work on. So thank you very much.
Thank you, Nikhil. I think a wonderful talk as usual. There are very few questions. You're still coughing,
so I'm so sorry about that. I'm also sorry that Max kept barking between me. So that is really unavoidable.
No. We didn't hear Max actually. This Cisco WebEx has a filter. We don't hear dogs actually.
Oh, very good.
But you were not well. But thanks for still putting in so much effort. We have had a good number of audience, and I think I'll give it over to Lopa. Lopa, you're muted. Lopa, if you have are there any questions you're muted, Lopa.
Yes, sir. There are 2 questions I could see in the chat box. First one is, which real-time PCR diagnostic kit is good to detect BCR-ABL?
It's a very difficult question to answer which kit is. So I don't get any money from like Colgate or Pepsodent or anything. So you can, whichever kit you use is good enough, provided you can make sure that the kit functions well. You are able to meet your quality control metrics, and you are able to pass EQUAS programs. So there is a fantastic guideline for CML which is published from the UK by Letizia Foroni's group. I suggest that anyone who wants to work on BCR-ABL testing, please follow this guideline. Make sure that all the quality metrics are met, and you will be fine. The kit doesn't matter. All kits that are used commercially originate from one paper which was published in Leukemia by the European Leukemia Net, which is the Europe Against Cancer Protocol, and all kits originate from that. So it doesn't matter really what kit you have. There is no magic here. Please follow standard quality control measures and guidelines. You should be fine.
Okay. So another question, is there any cell count like how to SAP ratio is there like that in BCR-ABL and control ABL gene?
Lopa, you have to repeat.
So the question is there any cell count cutoff like HER2 SEP ratio in BCR-ABL and control ABL gene? So I think this HER2 SEP ratio is for fish counting. It is not for real-time PCR where BCR-ABL and control gene cutoff is there. See, the BCR-ABL assay is really very sensitive. And once again, all the context when you diagnose disease has to be seen. So if you have a patient with a full-blown myeloid malignancy and you have one copy of BCR-ABL, it has no meaning because clinically this has to make sense. Your diagnosis has to make sense. So there is no standard cutoff like that. You have to, this is the test used to diagnose cancer, and it has to fit in with the clinical context of the disease that you're trying to diagnose. So there is no cutoff like this that you have for fish because false signals are not supposed to happen in the first place.
Okay. I cannot see any other questions from my other question. So any patient with cytopenia for how long, like when nothing is detected by regular testing like flow or any other bone marrow, when can we suggest for molecular diagnostics, sir?
So,
Any
so the,
cytopenias
inve-
or
so,
anything?
so the investigation of cytopenia is like this. Firstly, you have to explain why the patient has cytopenia. If you cannot explain it, then the next is, are you trying to then investigate the patient for MDS? For all MDS patients, NGS now is a standard investigation. There is no thing like first we will do morphology, then we will do flow, then you will do NGS, because all of these are subsequent tests in which you lose time waiting for a result. So the minute you think that am I investigating this patient for myelodysplastic syndrome, if the answer to that question is yes, you must send a sample for morphology, for cytogenetics, as well as for next generation sequencing. It is now a standard test. It is now no longer any hi-fi or advanced test.
Okay, sir. Thank you, sir.
Thank you. So Nikhil,
Yes, sir.
there are one more
one
question.
more.
Yes, yes.
Is there for monitoring Philadelphia like AL where in partner gene is ABL?
This is a very difficult question to answer. So can you answer? So let me give a bit of context here. So in PH like ALL, there is an entity which is known as ABL1 class rearranged BCR-ABL like ALL. These patients have translocations involving ABL1 gene, ABL2 gene, PDGFR-A, PDGFR-B, and the reason they're called as ABL classes, all of them can be treated with dasatinib, imatinib, nilotinib, what have you. So the problem here is that the ABL1 gene that is there can have several different partners. Like you have BCR-ABL1, you can have ETV6-ABL1, you can have you can have so many different translocations, and each of them then needs to have a different real-time PCR. Because PH like ALL is such a variable entity, it is not really possible to develop a new molecular test or real-time PCR test for this. So what the world is doing and what other people are doing to monitor this is, number one, you can develop droplet digital PCR-based tests which do not require a standard or a calibrator to monitor disease. Alternatively, instead of monitoring this fusion transcript, you can straight away do flow cytometry as well as the monitoring of IGH gene rearrangements by next generation sequencing. So in our lab, what we do is we monitor these fusions using next generation sequencing, or sometimes we are now start developing or standardizing these IGTCR based rearrangements for monitoring of this using NGS.
Sir, one more question. For somatic mutations, how much should be the minimum depth of NGS? For example, what would be the commonly missed in 100x depth?
So, how to answer these questions? So you must understand how next generation sequencing works. The way these next generation sequencing testing is done is by a statistical method. All variant callers have a cutoff of 5% or so, wherein they require a minimum number of alternate reads as compared to a reference read to say that this particular mutation is true or false. So even if you, so typically most NGS assays have a depth of, have a variant allele cutoff of 5%. But what should be the coverage? For germline testing, the minimum coverage that should be there should be approximately 80 to 100x, at least 50x for detecting a germline mutation. That means for BRCA testing or exome testing, the minimum coverage should be around minimum of 50x. For somatic mutations, the minimum testing should be more than 500x, at least 500x, if not 1000x. Okay, so for large gene panels, we have a 150 gene panel now in our lab, and for everything, the mutational minimum coverage would be at 700x. So 500x, if it is less than 500x, we just repeat the sample. So 500x should be an approximate cutoff for minimum depth.
Perfect. Nikhil, there's any more questions, Nishi?
No, sir.
No. Okay. Nikhil, do you have any final comments for students, for the audience here? You want to say something?
No, so I would like to say that molecular diagnostics is a field which is built upon knowledge of pathology. So people, I get a lot of people who want to just finish their MD and start doing molecular diagnostics. I don't think that is such a great idea. You must first build upon a specialty like surgical pathology or hematopathology, and then take on a super specialty field like molecular diagnostics. You can contribute and understand things much better. So and you are free to come contact us, me, sir, for if you want to learn more about this field.
That's very kind of you, Nikhil. Thank you. If there are no more questions or anything, Lopa or Nishi, you have any comments or any questions?
No, sir.
Thank you very much. I think with this we come to the end, and the next talk is third talk in the series of molecular diagnostics by Mayur Parihar. He is a faculty consultant hematopathologist, molecular pathologist at TMC Kolkata. On Thursday, he's talking on cytogenetics and cancer diagnostics. So don't miss. So we've had first talk by Rakesh on solid tumor, second talk by Nikhil, third is on Thursday, and all these three talks will keep on the replay till Sunday evening. So if you have missed out, so you have an opportunity to enjoy these talks till this Sunday evening, and please tell your other colleague residents who missed out on these fabulous talks. And next week, from next Tuesday onward, we're starting surgical pathology chapter, and the first chapter is head and neck pathology. And the first talk in that is by Dr. Katha Kante Labade. She's an associate professor at TMH Mumbai, and she's going to talk on crossing and reporting minimal data set of common head and neck tumors. So we have molecular talk coming up on Thursday on cancer cytogenetics by Mayur. After that, next week we're starting with surgical pathology and neck pathology. Thank you very much.
Thank you, sir. Thank you, Dr. Lopa and colleagues for this. Thank you very much. Bye.
Thank you, sir, for such an informative talk, sir.
Thank you. Thank you.
Thank you, Nishi, very much. I'm happy that you could join, Nishi. Good.
Yes, sir. Participants
Dr.
of
Nishi.
for his talk.
Yeah, no, fabulous talk. Always lovely listening to Nikhil. So much of to learn, and as he says, youngsters can come and apply for fellowship. He has 2 or 3 positions. He has PhD positions also. So it's not only fellowships. There are PhD positions also. With Nikhil, he's got 3 or 4 PhDs this year. So any youngsters, even MDs, want to join, you can get in touch with them. So thank you, everybody.
Thank you, sir. Thank you, sir. Bye.
Tata, everyone. Bye bye.
Bye.
I have to stop recording now.